Hello everyone, today I will introduce a review article, Diabetic Peripheral Neuropathy, Molecular Staging, Risk Factors, Therapeutics and Emerging Trends. This review focuses on diabetic peripheral neuropathy, or DPN, and summarizes its molecular staging, risk factors, current therapeutic strategies, and emerging intervention directions. DPN is a common and disabling complication of diabetes. However, its clinical phenotypes and underlying mechanisms are highly heterogeneous, which makes early recognition and precise intervention difficult. Therefore, a unified staging framework is needed to connect disease progression, subtype differences, and therapeutic targets. This review organizes the pathological progression of DPN into three continuous but interconnected stages. The first stage is metabolic dysregulation, mainly including hyperglycemia-driven biochemical toxicity and dyslipidemia-induced lipotoxicity. The second stage is chronic inflammation, characterized by neuroinflammation, pro-inflammatory cytokine activation, and microvascular dysfunction. The third stage is neuronal damage, which eventually manifests as small fiber or large fiber predominant neuropathy. Within this framework, the heterogeneous manifestations of DPN are further summarized into 6 representative subtypes: hyperglycemia-driven DPN, dyslipidemia-driven DPN, inflammation-driven DPN, dysvascularity-driven DPN, small fiber DPN, and large fiber DPN. Importantly, these 3 stages should not be understood as strictly linear or isolated events. Instead, they are connected by continuous pathological crosstalk. For example, early metabolic injury can trigger mitochondrial reactive oxygen species generation and oxidative stress, which then promote inflammatory activation. In turn, chronic inflammation and microvascular dysfunction further aggravate nerve ischemia, demyelination, and axonal degeneration. Therefore, redox disequilibrium is highlighted as a central hub throughout DPN progression. It links metabolic abnormalities, inflammatory amplification, vascular injury, and neuronal damage into a unified pathological continuum. Through this 3-stage and 6-subtype framework, the review provides a clearer mechanistic explanation for DPN heterogeneity and offers a conceptual basis for risk stratification, stage recognition, and mechanism-guided intervention. Current evidence indicates that DPN is not driven by a single factor. Instead, it arises from interactions among genetic, epigenetic, and metabolic abnormalities. Among these, metabolic factors are particularly important because they act upstream, shaping oxidative stress and the tissue microenvironment while amplifying inflammation, vascular dysfunction, and neuronal injury. Integrating these risk factors may support earlier identification and more precise risk stratification. Current DPN therapeutics can be organized into 3 major directions: metabolic management, anti-inflammatory intervention, and nerve repair. Metabolic management mainly targets early metabolic abnormalities. Anti-inflammatory approaches address chronic inflammation and microenvironmental dysfunction. Nerve repair strategies are more closely related to neuronal damage and functional recovery. Together, these strategies form a multi-layered and mechanism-oriented framework for DPN management. Beyond conventional approaches, cold atmospheric plasma, or CAP, provides a promising emerging direction for DPN research. Current evidence suggests that CAP may act through redox modulation and influence abnormal metabolism, chronic inflammation, and nerve repair. Therefore, CAP may serve as a potential cross-stage and multi-mechanism intervention strategy, although its clinical value in DPN still requires further validation. This figure summarizes the possible relationship between CAP and the 3 pathological stages of DPN. CAP may contribute to metabolic reprogramming in the metabolic dysregulation stage, inflammation attenuation in the chronic inflammation stage, and neurogenesis promotion in the neuronal damage stage. Its role is not to replace current therapies, but to provide a new research direction for cross-stage, multimodal. In summary, this review reframes DPN progression into 3 continuous stages: metabolic dysregulation, chronic inflammation, and neuronal damage. It further integrates genetic, epigenetic, and metabolic risk factors with stage-aligned strategies, including metabolic management, anti-inflammatory intervention, and nerve repair. CAP is proposed as a potential cross-stage, multi-mechanism direction for future DPN research. Thank you.